Okay, so this video will serve as a quick review of what we went over in lab about meiosis. So remember, meiosis has two goals. One is to reduce chromosomes, two is to produce genetic diversity. It's broken up into two steps, meiosis 1 and meiosis 2. So what I'm going to be talking about today, you can follow in figure 9.5 of your book and in figure 9.6. Many of the things that happen in these steps are very similar to mitosis, so I'm really just going to put up the highlights here, the things that are different. So in prophase 1, just like in mitosis, this is when the nucleolus disappears, the nuclear envelope disappears, the centrioles are going to move to their poles. But one very important thing that happens here is crossing over. Actually, we should probably start off with synapsis. So remember, this is where the homologous pairs find each other. And once they are next to each other, this is when crossing over can occur. And crossing over is when we can get some of that genetic diversity. So crossing over helps us to achieve that goal of genetic diversity. We talked in the last video, this is where the ends of two sister chromatids are going to switch with each other. So first off, we're going to have synapsis. Homologous pairs will find each other. Then in metaphase, the homologous pairs line up. So we did this simulation in class. And remember, they line up on that metaphase plate. So now we will have a set of pairs next to each other near that equator, in that line, so that in the next step, half of them can go one way, half of them can go the other way. So homologous pairs are going to align during metaphase. And then the homologous pairs will split. And so when they split, half of a pair will go to one side, half of a pair will go to the other side. So at the end of telophase 1, we have two nuclei. And then until interkinesis, we will end up with two cells. So we will get a division of the nuclei in telophase. The nuclear envelope will come back around them, and then we will divide the cells in interkinesis. So from there, we will then go to prophase 2. There is no synapsis because there are no more pairs. There is no crossing over here because there's nothing else going on. All we're going to have is the regular stuff from mitosis. So we'll have the nuclear envelope, will dissolve, we'll have our centrioles go to the poles, and we'll have spindle fibers attach, just like we did in mitosis. All these things are also happening in prophase 1. I just don't have enough room here. Now, since we don't have pairs anymore, we will have the chromosomes align on that metaphase plate in metaphase 2. So metaphase 1, it's homologous pairs. Now we just have chromosomes. We only have one of each. This was the division. Remember, we are diploid here, we are haploid here, so we only have half, the other point. Then in anaphase, sister chromatids. Each of these chromosomes, remember, is composed of a sister chromatid. Now they will separate. And so when we're done, this cell, which had one nuclei, will create two. This nucleus will now be two. We will now have a total of 4 nuclei. And then during cytokinesis, we will finish our cellular division. and we will have 4 cells. So mitosis will give me 2 cells. Meiosis, when we're done with it, will end up with 4 cells. And so, like I say, you can follow that in figure 9.5 and in 9.6, and we can see our meiotic division.